Hello and welcome to a day in the life doing a BSPP summer internship at the Crop Science Centre in Cambridge. The first thing I did today was head straight to the lab. I needed to set up a PCR reaction, and I like to do these first thing as I can leave them running and get on with other tasks. I am currently cloning genes involved in the plant immune response, so I need to use PCR to amplify the specific genes that I'm interested in before I can clone them. Here are my reactions in the PCR machine. I had a bit of spare time, so I looked at some sequencing results that just came in. Next I headed back to the lab to transform some cells. Here's me heat shocking them. Whilst my cells were incubating, I decided to have lunch. Then I plated out my cells and put them in the incubator overnight. I ran a gel for the PCR reaction I had run earlier in the day, and you can see an image of my gel. Since my PCR was successful, I could run a PCR cleanup on my samples so they are ready for the next stage of the cloning process. I tested the DNA concentrations on the nanodrop machine. Later in the afternoon, I started some cuttings for the 2 sweet potato lines. I then needed to water some plants. In another one of the growth chambers, I spread out some benthy plants and watered them. The last thing I did in the lab today was streak out some agrobacterium that I used for infiltration later in the week. In the evening, I went to a concert run by the Cambridge Botanic Garden. Thank you for watching, and I hope you enjoyed my day in the life.